Hello all, welcome to Vidhai. Exams Vidhai is a non-profit initiative by the students of IIITDM Kancheepuram to give free coaching and resources to students preparing for national level competitive examinations like NTSE and NMMS. So today our topic is heredity. First, let us check out what are the contents in this video you are going to learn. First, introduction, then pause and answer, then Mendel's work, sex determination, DNA and genes, mutation, and at last we'll sum up the topic. So first, few basic questions. Pause your video and answer them. Whom do you resemble in your family? Your mom, your dad, or your grandparents? How do you think color complexion, height, eye color, hair type are determined for a baby? What is DNA? What are genes? Our first topic is heredity. What is heredity? The transmission or passing of genetically based characters or traits from parents to their children is called as heredity. So what are these genetically based characters? They are nothing but height, weight, color complexion, etc. Then the next topic is genes. What are genes? So genes are nothing but they are the specific parts of chromosomes. Do you know where do these chromosomes exist in your body? Yes, they exist in your cells. Again, these genes will have two different alternative forms and those form a character. So each of which produces different effects in an organism. So these alternative forms are called as alleles. Example, in case of pea plants, the stem height is controlled by two alleles, one for tallness and other for dwarfness. Another subtopic is generations. What are generations? Take two parents. When these two parents are crossed to produce progeny, their progeny is called as first filial generation or F1 generation. When you take this F1 generation again and if you make them cross each other, then you'll produce a new generation known as second filial generation or F2 generation. Now we'll discuss about a scientist, Mendel and his works. So he has basically given us three laws: law of dominance, law of segregation, law of independent assortment. So laws of Mendel are them. And one more important thing is Mendel was known as father of genetics. What Mendel did is he selected a plant known as Pisum sativum or colloquially garden pea. So he used certain number of contrasting characters for garden pea. The characters you can see in the table: stem height, flower color, flower position, pod shape, pod color, seed shape and seed color. So as I've told, you'll have two alleles. One is dominant and other one is recessive. For stem height it's tall and dwarf. For flower color it's violet or white. Similarly, for other things we'll have axial, terminal, inflated, constricted, green, yellow, round, wrinkled, yellow and green. Pause the video and observe the table carefully. Observe this dihybrid cross. First, you need to take two different seeds and each seed has two different gametes. One gamete is capital Y, another one is capital R. For other seed, you have two such similar different gametes, small y, and small r. So in this P generation, you have two different seeds. When you cross them, you get F1 generation. So the output you get here is capital Y as one gamete and capital R small r as another gamete. Now you need to go to F2 generation. What's F2 generation? The output you get in F1 generation, you need to cross them once again. Now, the available gametes are capital Y capital R, small y capital R, capital Y small r, and similarly you get same gametes for other plant, for other seed I mean. Now you will be getting, when you cross them, you will be getting these many varieties. So the phenotype ratio is 9 is to 3 is to 3 is to 1. You need to remember this one. It's an important ratio. Many times you will be asked this question. Wait, what are these capital Y, capital R, small r? I'll explain you more briefly. Capital Y in the sense, look at this cursor. Capital Y in the sense, yellow color. If yellow color is dominant, then they have represented it in the form of capital Y. If the yellow color is not dominant, they'll represent it with small y in this textbook, remember. So, if yellow color is not dominant, you'll get automatically green color as seen here. Green versus yellow. So, if yellow is dominant, then they will represent it with the help of capital Y. If it's not dominant, small y. And coming to another, round and wrinkled. If that's round, then they will represent it with capital R in this textbook. If that's not round or wrinkled, they'll represent it with small r. Small r in the sense, round is recessive genes. As I have said, you will be having two types of genes, dominant genes and recessive genes. Dominant genes is nothing but which will be appearing from outside. Suppose each of us will be having a dominant and a recessive. If you take an example of a human's height has dominant and recessive. One is tall and other one is short. So if he's tall, I mean the height of a person is visible from outside, right? You can easily say if a person is tall or short. So if he is tall, it means the genes which is tall is dominant, and the short genes in his genes is recessive. I hope you got it. If he is tall, then it means the tall genes is dominant in his body. So this is all about it. As I have said, if it is capital Y, now look at this seed here. It's yellow in color. It means yellow color is dominant. So why they have kept it as capital. Now whereas here yellow color is not you can see it in green color. So it means yellow is not dominant. Yellow is a recessive genes. So small y. And round and wrinkled also can be appeared here. The same logic even suits to this. Now if you check here, you will be getting okay. Now in this table we have two different characteristics. One is color and other is shape. So if you look at the gametes here, so if capital Y, capital R, the first characteristic color. So color one is Y. So as there are two capital Y's, there are two dominant Y's only. So yellow will be appeared from out. Coming to the shape, round is again dominant genes. So you will get a yellow and a round shaped seed as an output. P as an output. Similarly, you can observe for other P's. Capital Y, small y. As dominant is present here, it will be showing yellow color. And round is again capital R. As dominant is R here, it will be showing capital Y, yellow color and round shape. Now coming to this B here, what happened here is it has two small y's. Yellow is not dominant here, hence it shows green color. Whereas for round shape, it has capital R and capital R. It has round as dominant, so it shows green color and a round shape. If you come to this again, the similar happen here. The color is yellow is recessive here, whereas The round, it has one dominant R, so it shows green color and round in shape. If you come to this, you'll get just one. 9 is to 3 is to 3 is to 1. In them, you'll only get this one as only one output. Small y. Both are recessive, hence it won't show yellow color. And both are again recessive for round shape, hence again it will be in wrinkle shape. So this is why we got 9 is to 3 is to 3 is to 1. It means we get 9 yellow and round piece and 3 just green and round piece and 3 yellow and wrinkled piece and 1 both wrinkled and green piece. So this is all about this. If you have any one dominant genes, the dominant genes will be appeared from out. So at least if you have one yellow capital Y, if you have at least one capital Y, then it will form yellow color. If you have at least one capital R, okay, let's go for another example. Let's take one gamete as capital T, capital T. Hence both has capital T's, tall is dominant. While here as both are small t's, the plant is dwarf. When you go for F1 generation, you'll be getting capital T, small t. Suppose you have got this one as one of your output, then again we are going for self-pollination here. Self-pollination in the sense taking the similar one, taking the same again and crossing it once again. So with that, you'll be getting capital T, capital T, capital T, small t, capital T, small t, and small t, small t. As I have told you, if you have at least one capital T, it will form tall. Hence here tall. Here also one capital T is available, hence tall. Same with here. While here you don't have any capital, so it forms short. So what is phenotypic ratio? Phenotypic in the sense you can observe it from outside. What you can observe if it's high, if it's tall or dwarf. So you have got three talls as the output and one short. Hence 3 is to 1 is the phenotypic ratio. Here it's a printing mistake, it's genotypic ratio. Genotypic ratio in the sense how many different genes you are producing here. Capital T as one genes, 1 is to, and capital T small t as 2. So 1 is to 2 is to, and small t as 1. So 1 is to 2 is to 1. So this is all about this. Hence we get phenotypic ratio as 3 is to 1 and genotypic ratio as 1 is to 2 is to 1. So the important terms you need to remember are phenotypic ratio and genotypic ratio. Phenotypic means the thing which you can observe from outside, like round or yellow, round green, wrinkle yellow. Such things can be observed from outside, hence they call as phenotypic ratio. So phenotypic ratio for this dihybrid cross is 9 is to 3 is to 3 is to 1. Genotypic ratio means the outputs you get for different types of genes, like capital RR, capital YY, you get 1. For capital RR, capital Y, and small y, you get 2. In such a way, you'll be getting a ratio 1 is to 2 is to 2 is to 1 is to 4 is to 2 is to 1 is to 2 is to 1. So these two are important ratios. You'll be frequently asked in your examination. 9 is to 3 is to 3 is to 1, and this long ratio. You need to learn that. So our next interesting topic is sex determination. What's sex determination? It means determining the gender of a baby or next generation. So for this sex determination, you have two factors. One is environmental and another one is genetic. Where environmental is observed is in some animals it is observed. How? It depends upon the temperature. When the fertilized eggs are kept in certain temperature, you get certain genders. Example is turtle. The next factor is genetics. So in genetics, example is humans. In us, animals like humans, Gender or individual is determined by the pair of chromosomes called chromosomes. So what are these sex chromosomes? So we humans generally have 23 pairs of chromosomes. Out of these 22 pairs of chromosomes are called autosomes, and the last pair or the 23rd pair is gender deciding chromosome and called as sex chromosome. This is one of the important question you will be asked in examination. Humans will be having 23 pairs of chromosomes, out of which 22 pairs are autosomes and the 23rd one is sex chromosome. So generally female will be having XX chromosomes and male will be having XY chromosome. And this is also another important question. XX is female, XY is male. Great. First, a question. Who is responsible for the gender of baby? Mother, father, or both? Yes, the answer is father. Why? As female have X and X chromosome and male has X and Y. Now multiply them. You'll get XX, XX, XY and XY. Only if you get XY, then it will be a male. So who is responsible for this? It's male. So the Y which is owned by a male parent is responsible for the gender determination. So remember this point. Father is always responsible for the gender of a baby, not mother. Because father has X and Y chromosomes while mother has X and X chromosomes. If you multiply them, you'll get either XX, look at this X, 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 or yx xy or yx again xy. So the chances are 50-50. If you get xx, then it's female. If you get xy, it's male. So how is this y forming? It's forming from the male's y chromosome. So it's all because of father. The gender of the baby is decided. Now, our very next topic is chromosomes. In nucleus of each cell. Where do you find this nucleus? In cell. So in nucleus of each cell, the DNA molecule. The DNA molecule is packed into a thread-like structures called chromosomes. So these are chromosomes where DNA molecule is packed into a thread-like structures. So each chromosome is made up of DNA tightly coiled many times around proteins called histones, and these histones and all you'll be studying in your higher classes. So that support its structure. So chromosomes constituency, let's check out them. They're made up of 50% proteins and 50% DNA. What's the full form of DNA? Deoxyribonucleic acid. Chromosomes are made up of many chromatin threads, each containing DNA and proteins. So somatic cells. What are these somatic cells? Somatic cells are the cells of your body except your sex cells or the sperm or ova. Except them, you'll be containing everywhere 46 chromosomes. While these gamete cells, it means the sexual cells or sperm or ova like them, they'll be containing just 23 chromosomes. Now 44 of our chromosomes are autosomes while 2 are called sex chromosomes. Each chromosome contains many genes that act as blueprint or set of instructions for the cell. To understand more about chromosomes, let us study first or let us know about the definitions of the subparticles. What's chromatin? The substance distributed in the nucleus of the cell that condenses to form chromosomes during cell division. It contains mainly of DNA proteins called histones. Means the substance distributed in the nucleus of a cell that condenses to form chromosomes during cell division. So the substance which is the reason for the formation of chromosomes is called as chromatin. What it mainly consists of? Mainly it consists of DNA proteins called histones. What's chromatid? A chromatid is one half of two identical copies of a replicated chromosome. Okay, now look at this diagram to understand more about this. So take this chromosome. So if you take it and if you check it carefully, you'll be having this small thread-like structures. So all this condenses to form this chromosome. So these are called as chromatin fiber. So what it consists of? Histones and DNA. So this is all about chromatid, and chromatid is this half of this chromosome which is the replica of other half is known as chromatid. So this half is called as chromatid, whereas this chromatin is the fiber which condenses to form chromosome is called as chromatin. And what centromer? The centromer is the constricted point at which two chromatids form forming the chromosome. So as I have told you before, the half of this chromosome is called as chromatids. So where these chromatids join, there forms a point known as centromer. So this is all about centromer, chromatid and chromatin. What's a gene? A portion of DNA molecule that serves as a basic unit of heredity. Genes control the characteristics that an offspring will have by transmitting information in the sequence of nucleotides on short sequence of DNA. A gene is a part of DNA made up of specific number of nucleotides. It is not a separate entity depending on the position of centromer. Different shapes of chromosomes have been identified. So look at this DNA. So in this DNA, this is all gene. So it serves as basic unit of heredity. It contains all the information and it will be transmitting the information in the sequence of nucleotides. So just now we have studied about centromers. Now with the help of number of centromers present, we will again further divide these chromosomes into subchromosomes or are different types of chromosomes like acentric without centromer, monocentric with one centromer, dicentric with two centromers, example wheat and maize, polycentric with more than one centromer, Ascaris and Lucella. And we have other special type of chromosome, polytene chromosome. So this is all about monocentric. Then holocentric have diffused centromer and microtubules are attached along the length of chromosome. Acentric chromosomes may break and fuse together to form a chromosome without a centromer. It cannot attach the mitotic spindle. I've told you before, without centromer, it's called as acentric. Then telocentric, rod-like chromosomes with centromer present on the proximal end, no P-arm or short arm present. Telomeric chromosomes are found in humans. Acrocentric, rod-like centromer present at one end giving rise to one very short arm and an exceptionally long arm. Submetacentric, L-shaped or J-shaped with centromer near the center of the chromosome giving rise to two unequal arms. Metacentric, V-shaped chromosomes with centromer in the middle giving rise to two equal arms. So these are the definitions. Now I will show their diagrams. You can compare them by pausing the video and reading the definitions. So first, acrocentric, metacentric, telocentric. The next topic we are going to study is mutations. Check out this: deletion, inversion, translocation, and duplication. So in deletion it's deleted, inversion it inverted, translocation it translocated, duplicated here for duplication. So these are the chromosome mutations. For duplication it duplicates the present. So the important chromosome mutations are deletion, inversion, translocation and duplication. So for deletion, it deletes the material. For inversion, it inverses. Translocation, you can see translocated. Duplication, it duplicates. Now we are checking out the functions of chromosomes. This is again an important topic. You may get questions from this topic too. So what are the functions of chromosomes? The main function of chromosome is to carry the genetic material from one generation to another. So the genetic material you are carrying now is from is carried since long many years from your ancestors to you. Now chromosomes. Chromosomes play an important role and act as a guidance force in the growth, reproduction, repair, generation process, which is important for the survival. Now chromosomes protect the DNA from getting tangled and damaged. Histone and non-histone proteins help the regulation of gene expression. Spindle fibers attached to the centromere help in the movement of chromosome during cell division. Each chromosome contains thousands of genes that precisely code for multiple proteins. So here you are introduced to the top layers, and the depth will be introduced to you in your higher classes. So these are the functions of chromosomes. So you need to remember them. So the main function of chromosomes is to carry genetical material or help an important or play an important role in guidance force for growth, reproduction, and all other things which are important for survival. And they protect DNA and spindle fibers attached to them in the sense to the centromere present in the chromosomes, which helps for the movement in cell division. And each chromosome contains thousands of genes that precisely code for multiple proteins present in the body. So this is all about functions of chromosomes. The next is about DNA. DNA is a carrier of genetic information from generation to generation. It transmits the hereditary in a coded language from parents to offsprings. DNA is a macromolecule or polymer. It is made of very large number of nucleotide units, and hence it is termed as polynucleotide. So it is nothing but the carrier of the genetic information from generation to generation. Means the genetic information that you are carrying now. It may be height, weight, or hair color, eye color, etc. and all. It's all from your parents. You're getting this, and to your parents, it's from their grandparents. It's from their parents. It means it's from your grandparents to them, and it's all about DNA. Now, each nucleotide unit in a DNA molecule is again made up of three different components. Deoxyribose sugar. It's one of the sugar. You'll be reading this in your intermediate too. It's a pentose sugar. So another one is nitrogenous base. Each nucleotide unit has a nitrogen containing base. In a DNA molecule, nitrogenous bases are of two types. First type is purines. The purines is in a DNA molecule are A, G, and pyrimidines are again in DNA molecule again C and T. What's A? Adenine. What's G? Guanine. C? Cytosine. And T? Thymine. You have four types: A, G, C, T. First, purines. In this purines, what are purines? Adenine and guanine. Remember, this is one of the important question you might be asked in your examination. Purines in DNA molecule are A and G, adenine and guanine. Next, pyrimidines. Pyrimidines are C and T, cytosine and thymine. And the third one is, after deoxyribose sugar, nitrogenous base. And this nitrogenous base you have studied about purines and pyrimidines. Purines are adenine and guanine, while pyrimidines are cytosine and thymine. Now the third one is phosphate group. The phosphate group contains one phosphorus atom and four specifically linked oxygen atoms. Phosphate, PO4. So what are the three components? Deoxyribose sugar. It's a pentose sugar, carbon molecule. Okay, next is CH molecule it is, and its next one is nitrogenous base. In this nitrogenous base you'll be having purines and pyrimidines. What are purines? A and G, adenine, guanine. Pyrimidines, cytosine and thymine.
Now the third component is phosphate PO4 group. So this is all about the nucleotides three components. Now look at this. G with C, T with A is compulsory. GCAT. So in this way all the things will be combined and this forms a DNA. Look at this. CG, GC, AT, GC. In this way it forms a DNA. So this is how your DNA is formed. The very next interesting topic is about genes. DNA on a chromosome occupying specific positions. For example, we have 30,000 to 40,000 genes on 46 human chromosomes. Each gene has specific sequence of nucleotides. You can see here how these nucleotides are arranged. So in this way each nucleotides will have specific sequence which determines its functional property. So it is the genes which determine everything about us, say your appearance, your color complexion, the shape color of your eyes, hair color, the shape of your nose, chin, and is it to be fat or is it to be thin, whatever it all, all of these are determined with the help of genes. Gene is a segment of DNA molecule which has coded information to form a particular protein in the cell. This protein can function either as structural protein or as a functional protein. Now we will discuss about mutation. Chromosomal mutation. What's chromosomal mutation? It discusses about the sudden change and mutation in the sense change, the sudden change in occurring chromosomes. So what changes can occur? First change is structure. Changes in structure of chromosomes. Structural changes in the chromosomes usually occur due to the errors in cell division. Changes in the number and arrangement of genes takes place as a result of deletion, duplication, inversion, translocation in chromosomes. We have seen this before itself. Deletion, duplication, inversion and translocation in chromosomes. So these are due to the change in structure of chromosomes. So the other one is change in the number of chromosomes. So these two are chromosomal mutation. It means change in the chromosome structure. First one is structure and the second one is number. They involve addition or deletion in the number of chromosomes present in cell. This is called as ploidy. There are two types of ploidy generally. First one is euploidy, second one is aneuploidy. We'll discuss about them now. About euploidy and aneuploidy. Euploidy is about all about addition, while aneuploidy is all about deletion. So I mean subtraction. So euploidy is the normal human or any individual. What they have is diploid number 2n. Usual number is diploid number 2n. If any species contains more than 2n, it's called as euploidy. So generally there are triploidy and tetraploidy 3n and 4n. They are generally greater than 2n. If the 2n is called as usual number, that's normal one. If they are more than 2n, I mean if they are 3n or 4n, then it's called as abnormal. So if they are in that way, then it's called as euploidy. So 3n, first let us discuss about 3n. 3n, what they'll be having three haploid sets of chromosomes, and these three triploid plants and animals are typically sterile. Sterile in the sense they cannot further go for reproduction. Now about haploid set of chromosomes. These triploid plants are advantageous because they often result in increased fruit and flower size. Now aneuploidy, as euploid is addition, I mean it's having more number. In haploid it has less number, the loss or gain of one or more chromosomes in a set. It has three types: monosomy 2n minus 1, trisomy 2n plus 1, and nullisomy 2n minus 2. In man, Down syndrome is one of the commonly known as aneuploid condition. You'll be learning all this in your higher classes. So one of the important question here is Down syndrome is due to aneuploid condition. Down syndrome is occurred in mans. So the three types of aneuploidy are monosomy, trisomy, and nullisomy. And remember these numbers: 2n minus 1, 2n plus 1, 2n minus 2. Now check out this diagram. First diploid. Check out this. 1, 2, 3, 4, 5, 6, 7, 8. So it's a 2n1. Here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So it's triploid, 3n. So you have three types times of n. So it's triploidy. And here it's 2 times of n, 2n. So it's diploid. This is about euploidy. And in aneuploidy, the diploid is here it's 2n plus 1 and it's 2n minus 3. So this is about aneuploidy and this is euploidy. 2n, 3n, 2n plus 1, 2n minus 3. Do not confuse, n is a natural number. Thanks for watching the video.